So our next subtopic is methods of determination of overall proteins. So here we will learn how overall proteins they are measured, they are determined in the sample. We will discuss it in different steps, and the method which we will discuss is Geldo Method. As the name indicates, this method was developed in 1883 by this scientist whose name is Johann Geldo, and it is a standard method for determining the total protein estimation in the sample. What is done here? A sample is digested with a strong acid so that it releases nitrogen, which can be determined by a suitable titration technique. So, जो नाइट्रोजन की से हम प्रोटीन एक्चुअली estimate करते हैं by the nitrogen concentration. Protein amount is calculated from the concentration of nitrogen by using a conversion factor, and this conversion factor is different from each of the protein. So हर ज़ाहिर है जितनी उसकी नाइट्रोजन वैल्यू होगी उसके हिसाब से उसमें प्रोटीन चाहिए मौजूद होगी. Each protein may have different F value depending on their amino acid composition. So if there will be more amino acid, there will be more F value, and if number of amino acids are less, then there will be less F value. इसमें जो different steps होते हैं वो हम one by one discuss करेंगे. First of all, digestion. तो इसमें क्या किया जाता है? Sample is digested by heating the sample in the presence of a strong acid like sulfuric acid or hydrochloric acid. And by this way, what happens? It converts nitrogen in the sample into CO2, into carbon dioxide, into ammonia and water. Ammonia gas, which is formed here, that is not liberated as it is, because ammonia which is liberated, that is in the form of ammonium ion, which binds to sulfate ion and remains in the solution. And similarly, this ammonia that also reacts with boric acid, and ammonium borate is formed, which is titrated with sodium carbonate solution. Next step is neutralization. What is done here? The solution in the digestion flask is made alkaline by the addition of sodium hydroxide, which converts ammonium sulfate into ammonia gas. Then it is liberated from the solution and moves into the receiving flask, which contains boric acid. And here, this ammonia that reacts with boric acid to form ammonium borate. Low pH converts ammonia gas into ammonium ion and simultaneously converts boric acid to the borate ion. And for here, different catalysts, they are used like mercuric acid, or sometimes other catalysts, they are also used. Next step is titration. Here, nitrogen content is estimated by titration of ammonium borate formed with some standard acid like sulfuric acid or hydrochloric acid, and a suitable indicator is used which indicate the endpoint of the reaction. Hydrogen ion concentration required to reach the endpoint is equivalent to the concentration of nitrogen in the sample, and this nitrogen concentration is converted to a protein content using a suitable conversion factor which we have discussed earlier on. And this varies for each protein. Here we can see in the left side the sample is heated in the presence of acid, and then in the right side we can see that this round bottom flask, then this ammonium ion is produced, and then on the further right side in the flask titration is going on, and where this titration is carried out in the presence of indicator which is used to find out the end point, and then this nitrogen concentration is calculated, and then we find out the conversion factor, and in this way protein is calculated from the conversion factor. Now, what are advantages of this method? This method has high precision and good reproducibility. There are different disadvantages with this method as well. It does not give a measure of the true protein of the sample, as all nitrogen in the food is not in the form of protein. ज़ाहिर है चूँकि इसमें नाइट्रोजन से हम प्रोटीन एस्टिमेशन करते हैं, तो जो फूड के अंदर सैंपल के अंदर नाइट्रोजन होती है सम टाइम दैट इज नॉट इन द फॉर्म ऑफ प्रोटीन, दैट मे बी इन द फॉर्म ऑफ सम अदर compound like some nucleic acids, they also have nitrogen, and similarly some other compounds they also have nitrogen. So इसमें ज़ाहिर ये डिसएडवांटेज है कि जो नाइट्रोजन की वैल्यू के ऊपर, नाइट्रोजन की कंसंट्रेशन के ऊपर जो प्रोटीन कंसंट्रेशन फाइंड आउट होगी, जाहिर है वो इतनी रिलायबल हम कहेंगे वो नहीं होगी. And then different proteins, they may need different correction factor as we have discussed earlier on, कि उनके अंदर जो अमाइनो एसिड्स होंगी अगर कम होंगी तो उसमें एन वैल्यू ज़्यादा होगी, अकॉर्डिंगली डिफरेंट कन्वर्जन फैक्टर होगा. अगर अमाइनो एसिड्स कम होंगे तो एन वैल्यू कम होगी और इस तरह डिफरेंट कन्वर्जन फैक्टर होगा. एट द सेम टाइम यूज ऑफ कंसनट्रेटेड सल्फ्यूरिक एसिड एट हाई टेम्परेचर has associated risks. So ये भी हमें ज़हन में रखना होते हैं. Different catalysts which are used in this reaction, they may also be harmful, and this process is also time consuming. So there are different disadvantages which are associated with this method.